Okay, stems. We've been looking at stems. Dicot versus monocot. So we're talking about the angiosperms. And we talked about how it's kind of weird that the dicot stem is showing this ring of vascular tissue, whereas in the roots, it's that plus configuration smack in the middle of the root, where you have the xylem and phloem, and now you see it morph into this ring of vascular tissue. So the bigger cells that you're seeing that are kind of, to my eye, they kind of look purpley red. Those are the xylem, consisting of vessels and tracheids. The xylem are the big cells, and then the phloem is the delicate kind of blue-green right to the outside of that. And so interesting shift in where you find the vascular tissue compared to the root tissue. And then on the right, you're seeing the idea of the monocot stem, where instead of any like definitive pattern, you just have this scattering of vascular tissue in small little bundles. So we'll look at that as well. So with the dicot, if you work your way from the very outside of the stem in, what you see is the epidermis, and then the cortex region is pretty much parenchyma cells that you see, and then you see the vascular bundles in that single ring. And then I'm going to talk about this vascular cambium idea. So we're going to see that that's right in between the xylem and the phloem, and it's a ring of cells that goes all the way around, and it will lead to secondary growth. And then the very center is what they call the pith region of the stem, which again is more parenchyma cells. Okay. All right. So when you think about a dicot stem, this vascular cambium is really, really important because it is an active area of growth. So it's meristematic tissue. It will keep growing and dividing through the lifespan of the plant, pretty much, if conditions are good where they can do that. And it's a ring of cells. And so it's right in between the xylem and the phloem. So I'm going to try to, so it's right in between, and it, whoops, forms a ring all the way around. So if you just follow that all the way around, and so what's going on there, this ring of cells, this vascular cambium, when it grows, when it goes through cell reproduction and produces two cells, they're two very different cells. One cell is a xylem cell, one cell is a phloem cell. So when it divides, I like to think of it how it produces, xylem to the inside, phloem to the outside, and it just keeps doing that over the span of the lifespan. And so what that's doing is allowing for that stem to get bigger and bigger in diameter. And so this so-called secondary growth is meaning that you get diameter growth rather than apical growth where things are growing taller and longer. Make sense? Okay.
Is that where the growth rings come from?
Yep. Yep. So this is a very young, we'll look at some other ideas here where they get older. But first, let's think about the monocot. They are very different because you're going to see an epidermal layer. The very outside surface is called the epidermis. And then you see all those scattering of vascular bundles. And there is no vascular cambium. Nowhere in here is there vascular cambium. So that means monocot stems have no significant way over the lifespan of the plant to get bigger in diameter. That's a huge difference compared to dicot angiosperms. So monocots, they don't have a vascular cambium. They cannot keep increasing their girth. They just don't. Okay. Here's a mock-up of the stem tissue. Most of it anyways, and then the very bottom row is actually what you see under the microscope. And what we see, we're going to get into this later and this one later. So you'll come back, you'll see the slide again. But because those are seedless vascular plants, we're not going to talk about them just yet. So we're looking at this idea here, okay? And then this, so the monocot's on the far right. And then there's kind of some variations to the dicot. They call it a U-stele. But basically it's this idea of a ring of tissue. And so when you look at the monocot, that vascular bundle, if you zoom into that, then this is a mock-up of it, but doesn't that look like a face, a surprised face? And they can look like that under the microscope. It's amazing. And so what you're looking at, the eyes, the nose, the mouth, those are all the vessel type xylem. Okay, those are the big vessels. And then the cheek area is the tracheids. So smaller xylem, right, tracheids. And then the forehead is the more delicate phloem. And so that's what you have pretty much going on. When you look at the monocot, here you don't really see a face per se. This is in the family, the lily family, but you can see how it's all scattered. Here's a really nice image. This is bamboo. So this was entered into it. Every year there's a contest, a small, what do they call that? Small things contest or something like that, where it's microscopy work that they enter into the contest. So you get this scattering of the vascular bundles. So no increase in diameter due to a vascular cambium. Here is a drawing trying to show a little bit more carefully where the cambium is located. And so you can see the pink is all the xylem, and then the green is the phloem, and then the red line in between is the vascular cambium. So the artist is correct. You know, I just got done telling you when the vascular cambium divides, it produces xylem to the inside and phloem to the outside. So if you follow that logic, you would think it would have the same amount of phloem cells as xylem, right? But you're not seeing that. So what happens is the older phloem starts to die. Those are living cells, especially when you start getting into the age where the stem is producing bark tissue. There's only so much that the phloem can take in terms of the pressure. And so the older phloem actually breaks down. The newer phloem stays good for, for the season. And so that's why you do see this lopsidedness. You see plenty of xylem, and those are the growth rings. Those are the annual rings or the growth rings. And so it looks like we have what? 1, 2, 3, 4 years of growth. So this plant is about 4 years old, it looks like. Okay, so you can count the xylem, the growing seasons. So there were 4 summers there, if it's a deciduous plant. And the more delicate phloem doesn't persist. It gets crushed essentially with more growth.
So then does it just become like supporting tissue?
It literally breaks down. Yeah.
Does it reuse the nutrient?
Yeah, those get siphoned into other cells, and plants are pretty good at scarfing up nutrients and making use of it like that. And so it's smushed right between the xylem and the phloem, and we call it secondary growth, where you have xylem cells as those cells are reproducing. You get 2 new cells. One will be the xylem and one will be the phloem. And so we call that secondary xylem, that's what you would call the wood if you're a forester. But technically it's secondary xylem. That's what you see when you look at those growth rings. And not all of the xylem is necessarily active. For sure, the most recent xylem is definitely active because that's the season's growth, that season. The older xylem tends to become less functional for transport, but it's certainly good for structural stability. And remember, xylem has lignin, so it's good, strong portion of the stem. And then we're going to see how really, when we think about bark tissue, when we get into that, phloem is actually part of the bark tissue. The way we define what bark tissue is, phloem is actually part of it. And so you can get, over the years, you'll get lots of those so-called tree rings, the growth rings, and that's the xylem that you're counting, but you don't see a whole lot of the phloem. And you got to be careful, like especially like birch trees, it's when they have the hanging bark and it's so tempting to pick at it and strip around like that, and don't do that. Does anyone know what that is if you strip bark all the way around in a circle? There's a word for that, what you're doing to the tree.
Girdle?
Yeah, you're girdling the tree. That's a big no-no. And when I used to walk in the woods when my boys were young, it's very tempting to pull on that flap of bark, birch bark. But don't do that all the way around, because if you take enough of the bark tissue off in a whole ring like that, all the way around, you're taking off the phloem, which means you essentially have sentenced that tree to death, because now the sugars that are being made in the leaves, as they course down the stem, they're going to reach the part that's girdled, and the sugar can't go any further. It's done. And so you can actually see with a girdled tree that there'll be a swelling right above the girdling, not hugely dramatic, but you can see it. And that is like the sugars piling up, so to speak, in those cells, and they can't go any further. So then the roots die. And if the roots die, of course, they're not supporting the above ground. They can't get water up there. So it's a mess. It's a slow death. Yeah. So don't do that, even though it might be tempting with the birch trees, especially. So be careful. So here's a real microscope slide of, it looks like 3 growth rings on this one, 3 years old. So you have 3 growth rings. So you can see different, so all the xylem, you can see some bigger vessels, some smaller tracheids. And then the vascular cambium is this dark blue line that you can see demarcating the 2 areas. So it's nicely colored here, where you can see the xylem very clearly separated from the phloem. And then this is developing bark. A lot of this is some bark tissue too, that we'll get into. So the vessels are the bigger ones in the xylem area, and the tracheids are the smaller ones. So you can actually look at that carefully and get a feel for what kind of growing season this plant might have had. And so if you see growth rings that have a lot of vessels in them, the bigger trachea, the bigger xylem, sorry. So if you see, like, that's a pretty good swath of vessels all the way around, that is the so-called spring vessels, they call it. So hopefully when spring comes around, the trees are sensing lots and lots of water in the soil. And around here it'd be with snow melt, of course, and the rivers and lakes filling up and have lovely wet soil. So if that's the case, and they can sense that, of course, then they're going to make vessels. They're going to lay down and make a lot of vessels over tracheids, because vessels take up water better. So if there's lots of water available, they'll produce vessels. And then if they undergo any kind of drought or even modest drought, they'll switch over to making tracheids, because tracheids they're smaller and they're not quite as efficient as vessels. But the one good thing about tracheids is if you make a smaller diameter for water uptake, you run less of a risk of an air embolism ending up in the xylem. I'll talk a little bit more about that. So they can actually decipher how much water they have to work with, and they can decide, should I make vessels or should I make tracheids? And that's why you see this differing pattern. Usually as the summer goes on, they switch over to tracheids. This, you can see a lot of what looks like vessels, really. And rather than there, it looks like some tracheids might be showing up here. So no, I think those are all tracheids actually.
Even on like the very left.
Yeah. I'm gonna double check that. But you can see, and now this one is forming bark. So the really dark outer portion is, we got some definite bark showing up there. Yeah, in my notes, they claim this is a ginkgo stem. This is taken from the textbook I was using. And ginkgos, believe it or not, I don't know if you have seen ginkgo leaves, they're really quite beautiful, so the fan-like leaves, but they're gymnosperms. They're gymnosperms. They're not angiosperms. And so gymnosperms don't have vessels. They only have tracheids. But these are pretty decent tracheids for tracheids. They're pretty big. But technically, gymnosperms, any plant that belongs into the gymnosperms, they're more primitive, and so they should only have the tracheids, not the vessels. The vessels didn't come about until the angiosperms. Okay.
Besides the
Mhm.
size difference, is there a structural way to like see? Because if we didn't know this was a ginkgo, I would have said the ones.
Yeah.
To the best.
Well, it fooled me at first too.
Is there a way to tell then? If it's just
Yeah, this is what clued me in is that it was so uniform. Like these really aren't that much smaller than these. So usually tracheids are noticeably smaller in diameter, like really different. And that's what was kind of cluing me in. And then I looked at the notes section and it's a ginkgo. And ginkgo doesn't have the vessels. So yeah, the spring vessels are spring wood, and then the summer wood, they switch over to tracheids. So I'll talk a little bit more about that as we finish up the idea on, we gotta think about transpiration a little bit more. Okay, and then the very center, that tends to be deeply pigmented, often it's very deeply brown. That's what they call the heartwood. And it's colored that way because the plant is usually laying down a lot of tannins and gums or resins. And that area is definitely no longer being used for water transport. However, having that section impregnated with tannins and gums and resins, that's very those are preservatives essentially. And so it makes it more difficult for fungi and bacteria in particular to gain a foothold in there and cause problems. So it's good structural integrity. They beef it up with these preservatives. So they're trying to minimize any of the pathogens gaining a foothold. And then they call the sapwood, those are the active areas. Though foresters often use these terminologies. And so that's the active xylem. They'll call it the sapwood. When I was a grad student, I rented it was essentially a mansion. It was this beautiful mansion on top of the hill leading up, or no, as you go through Hancock. Has anyone been to Houghton Hancock? And you go around and up on the highway, there's a mansion up there called the Quincy Mansion, and that's where I rented the servants' quarters. I lived in the servants' quarters, which was awesome. But anyways, it was like a 10-bedroom home with 2 kitchens, one upstairs, one downstairs. And it was actually built for the manager of the mine, the Quincy mine. And there used to be a tunnel. So the mine was right next to it. And there used to be a tunnel that would lead from the basement of the mansion to the mine. So he wouldn't have to go out in the winter. And so they built this huge mansion to try to entice him to come. He was living in New York and they needed a manager for the Quincy mine. And so they built this beautiful mansion. It's incredible. I can't believe grad students rented that thing. I mean, it was this gorgeous mansion with antique furniture in every room, and they let us. I'm kind of amazed the landlord at the time was okay with that. But anyways, we were respectful, but that was kind of a risk. Where was I going with this?
Funnel.
Tunnel. Foresters, sorry. So I roomed with a bunch of foresters. So the landlord who owned that, the person who owned that house at that time was a geologist, retired geology professor, and he had a soft spot for forestry and geology students. I don't know how I got in there. I was biology. I snuck in there somehow. But so anyway, so I would talk a lot about the wood cuts and the different, and they would all use this terminology. And so this is very common in the forestry world to use these types of terms. Here's a nice, so the pith area, that's just parenchyma cells. Sometimes that hollows out and those die out and you'll have a hollow core. Depends on the plant. Here's a really good image of the spring wood versus summer wood. You can clearly see the difference. And so wherever you see the vessels, that was the spring growth, where they had plenty of water. So if that area of the tree ring is really wide, you know they had a really great summer. Lots of precipitation and they got to grow really well. And then in this case, though, we don't really see that. We see a pretty modest ring of the vessels and then a big long swath of tracheids. So that, to me, I would interpret that to be a pretty dry summer overall. Short spring, not much rain. Not much rain in the spring, I guess is what I mean to say. And then kind of a drought period through the summer. So once you know a little bit about that, you can actually learn about the life of the tree, like what did it experience over those different summers. Rather than just counting how old it is, you can actually say more than just how old it is. So then pine trees, they're gymnosperms. So again, they don't have the vessels. And so when you look at a cross section of a tree trunk of a gymnosperm, like red pine, white pine, so on, you should see much more uniform tracheids is what you're seeing. Okay, and they pretty much are the same size. There's a little bit of variation. But if you look at that compared to that, big difference. So the angiosperms, they have this ability to choose what they want. Do they want the vessels or the tracheids? Here's the heartwood versus the sapwood idea. And then you often hear people talk about hardwoods versus softwoods, if you've ever wondered what that means. Usually when they say the hardwood, they mean dicot trees. Okay. All right. So let's think a little bit about bark. It's got some complexity to it. We're going to see that there's another ring of meristematic tissue to help build the bark. And it's super good at protecting the stem overall. And so it's going to be built up with waxes. And the definition of bark is that second bullet. All tissues outside the vascular cambium is bark tissue. And since we know that vascular cambium produces phloem to the outside, that means phloem is part of the bark tissue, all right? And so secondary phloem is part of the bark, and then the periderm is part of the bark. This is kind of when you get older, when the tree gets older, it will shift over from the having that epidermis that it grew up with. It'll get replaced with the so-called periderm. So when you look at the periderm under the microscope, you're going to see several layers. There's the cork cambium, and I put in parentheses these weird terms, phytogen. I don't use those, but you're gonna see some slides with these words on them, so I focus on the word or the phrase cork cambium. So that is a meristematic tissue. So like the vascular cambium is meristematic, that gives rise to the xylem and phloem. The cork cambium gives rise to cork cells to the outside. So it's another ring of cells, and when it divides, it's going to make cork cells to the outside. And they call it secondary cortex cells to the inside. Those are just parenchyma cells that will be produced. So it's interesting that there, again, this cork cambium is going to produce one cell that ends up being dead, just like with the vascular cambium. We said, xylem to the inside, the xylem dies. Well, with the cork cambium, again, you have the cork cells that will be produced. Those will die at maturity, and they get really heavily impregnated with suberin, which is a wax. And then the cells to the inside of the cork cambium are the cortex cells, our secondary cortex, just parenchyma. Those are living. And of course, they gave it another term. I don't use the term phytoderm, but you're going to see that on some slides. So I want you to learn parenchyma. So bark is, there's no like uniform way other than we know about these layers like this and that there's a cork cambium and all that. But when you look at the actual bark from the outside of the tree, it's so incredibly varied. And you have different thicknesses, different roughness. It's really, there's no easy way to define it in that regard. And there's the birch idea with the hanging strips there. This is bark too, believe it or not. That's not photoshopped. That's the real colors. Rainbow eucalyptus, you got it. Isn't that fantastic? Have you seen one with your own eyes?
But I want to.
I know, me too. I wish they could grow in our greenhouse, but I don't think they'd make it. These are very tropical. I think they're my friend Kim, she goes to Hawaii a lot. They're in Hawaii. I think they're in Australia and New Zealand as well.
Was-
But what I think is so fascinating is that's not Photoshop, that's the real colors. And then the bark will peel off and reveal different colors over time. So it's like this ever-changing color, rainbow colors going on. So, but I don't think it would really grow in our greenhouse, especially when we end up not having heat. We've had that issue. When was that? Just a few weeks ago. The heat went out in the science building and the greenhouse temperatures plummeted to 42 degrees. So I think this tree would keel over on that. But I love it. Got to see one before I leave. All right, so and then you go to something like this. It's pretty, generic looking, but this one's really thick. Lots of barrier, lots of waxiness. So here you're seeing kind of like this growth. The upper image is younger, and then the lower image, it's a little bit more growth. So they're just trying to show how how those layers kind of build up over time. So the brown are the cork cells. So those, and they do look like that under the microscope. So those will die at maturity. And then the cortex, and those are just parenchyma cells. Okay. Here's an actual picture. So what you have is, in this particular image, you have a lot of cork cells. So all of that brick-like swath. So this whole region looks like cork cells, and then probably all the way through here. And then you have the cork cambium in a ring, and then the parenchyma cells. Here, this one's nicely labeled with those old terms. So the cork cambium. So the cork is to these are all dead cork cells. to the outside of the cork cambium. And then you have the parenchyma cells to the inside. So when you look at a more mature tree trunk, you actually have two rings of cells that will be producing new xylem and phloem in the one more inner portion, and then you have the cork cambium that will be making more bark tissue. So here is a nice depiction showing both. So you have the vascular cambium is separating the xylem from the phloem. So it's right there. Whoops. And then the cork cambium is a little further out. So two rings of tissue that are actively growing through the life of that tree. And the vascular cambium is devoted to xylem and phloem, and then the cork cambium for bark tissue. And so they're trying to show everything to the outside of the vascular cambium, which means the phloem. That is part of the bark definition. Okay, so the bark on the trunks of trees is very well designed for protection, very hydrophobic. You know, when you think about all the water that's coursing through the xylem and the minerals dissolved in it, and then the sugars coursing through it, it's pretty attractive for, you know, especially bugs to want to get in there. That's some good nutrients to get after. So it makes sense that the bark tissue is beefed up to try to protect against that. However, the living tissue in the tree trunk does need to breathe in the sense that it needs oxygen so it can do respiration. So the lenticels, that's the job. We briefly mentioned those during the first or second week. And so again, with lenticels, it's just a looser patch of bark tissue. And sometimes it's very, very noticeable, like on the picture on the right. Those are obvious bumps that make you go, huh? So those are the lenticels. And it's just because of the more loosely packed cells, it does allow better gas diffusion through there. And primarily what the tree trunk wants is the oxygen so that the oxygen can get to the living cells. And it's part of the cork cambium process that happens through that action. But then down here, you can see the apple and pear. And if you've ever wondered what those little dots are, those are very dainty lenticels on that. So the skin of the apple and pear is pretty protective and waxy, but it still needs the flesh underneath that's living, needs that oxygen. And so if you've ever wondered what those spots are, that's what that is. So not all tree trunks have really noticeable lenticels. Some do. I mean, these are pretty noticeable. But that other image I showed you, where is it? Like, I couldn't tell you where the lenticels are on that one. I wouldn't know. You'd have to like take a section and look at it under the microscope. But those are more noticeable. And then under the microscope, you see that loosely packed region right along the perimeter there. So it's a good strategy. It's still minimizing risk because it's discrete regions. So yes, you are opening yourself up to maybe some insects trying to get it in through that area, but you still need oxygen. So it's kind of you have to get oxygen somehow. And then with birch, you often see those streaks like that. And so it would be more like that for your lenticel. Stem tissue can be modified into different looks and purposes and uses, functions. So let's think about some. So on the right is a beautiful giant saguaro cactus. So most of that stem tissue is storage. What are they storing? Water, lots of water. And all those lovely spines to try to protect it. The thing you're seeing on the bottom, those are flattened stems. They look like leaves. So it's easy, it's understandable why you might think those are leaves, but those are stems that have been flattened out into that morphology. And the fancy name is cladophyll. So cladophylls are flattened stems filled or covered with spines. Next week in lab, I think you'll be looking at some modified stems. So rhizome, if you think about a rhizome, the way I define it, that is an underground stem. Underground stem. The tuber, like a white potato, the tuber is an underground stem that's been modified into that tuber shape, and it's chock full of carbohydrates and other good things, okay? So you'll look at some different rhizomes. So a tuber is a rhizome. So that means a tuber is an underground stem that's just been, its morphology has shifted a little bit into that tuber shape. So that means when you eat white potatoes and you keep the skin on, the skin is actually bark. So you're eating bark tissue when you eat you should eat the bark, right? That's a really healthy area. So then you think about like ginger rhizomes, we'll look at those. And then the cladophyll, we don't really have any cladophyll, I don't think, to show you, but we'll have some bulbs. So bulbs are more leaves than stem tissue. So the stem tissue is very modest, and you have more leaf tissue. And then the corm is the way to, like the opposite. A corm is really, when you look at the whole thing, it's mostly stem tissue, and the leaf tissue is just those papery, dead-looking things that surround the corm. And then we can have modified stems that form tendrils or runners. So stem tissue can do a lot of different things. Here's some actual images. So what is a tendril trying to do? What's the purpose of a tendril?
Find things to latch onto.
Yeah, find something to latch onto. So you're usually thinking about vining plants that don't devote a lot of strength to any, like a tree trunk, and they are relying on something to latch onto to keep their leaves up off the floor, forest floor or whatever, and get access to sunlight. Then you can have plants that are like, they look really squished, top down, where their stem is super short, and all the leaves tend to attach almost at the same spot around that short stem. So we call those rosette plants. There's plenty of plants that do that kind of pattern. And then hawthorn thorns, so some stem tissue can morph into thorns for protective function. We'll look at ginger rhizome in lab next week. Iris is another type of rhizome. And then the tuber. So this is bark tissue that you're looking at. So all these little streaks or dots, those are lenticels. Okay, that's actual bark tissue. And then you can see the top right one is sprouting. So if you look at the bigger areas like that, those are the node regions of the stem. So it's a bloated stem, but it still has nodes, which means the node region is active areas of growth. And so you can see it's starting to sprout. So if you plant that in the ground, those areas will grow into stem and leaves above ground. Okay, so that's how they're called seed potatoes. Ever heard that phrase? They're called seed potatoes, even though they're not seeds. They're actually tubers that you buy. And then what I need to do to save money is I'll buy seed potatoes and I'll look to see where the nodes are, and then I'll cut the seed potato, making sure that each cut part has a node. And then you plant those cut pieces and they'll get you. So out of one tuber, I can often get 5 to 8 plants out of one tuber. So as long as it has a node, it will sprout. So you eat bark when you eat that. Would you eat that? So this is potato scab disease. This is what I studied on my postdoc. This is caused by a pathogenic bacteria. And it looks disgusting. It looks like scabs. Doesn't it look like when you scab your skin? That's why they call it potato scab disease. It looks nasty. You don't really see that in stores. Very rarely will I find any scab on potatoes in stores. This is usually a total loss to the grower. They have to either feed it to their pigs if they have some, and sometimes even the pigs won't eat it because it doesn't taste good. When I was studying it, we had a potluck dinner where every meal had to be made from potatoes. So all we ate was potatoes, and then my postdoc advisor brought out some scabby potatoes and dared us to take a taste, which we all did, and it tastes like how wet dirt smells. So it tastes like you're eating wet soil. So it's not tasty, but it's not dangerous. She knew that, this pathogen does not interact with our bodies at all. It only likes potatoes and related plant species. So you don't want that showing up. So when you dig up your potatoes, if you get that, don't eat it. It's not going to taste good, but it won't hurt you. It won't make you sick. It just tastes awful. Corms and bulbs. So they're kind of the opposite of each other in terms of the stem tissue amount. And even the leaves, like onions, have very fleshy leaves, whereas corms tend to have very thin, papery leaves. So what we just looked at, I kind of put it onto one list. So if you are studying this, sometimes looking at lists can help as you, put it all together. Okay, so when you're looking in the book, there is sections in each chapter on the roots and stems and leaves, like uses of, like how do we use roots? How do we use stems? How do we use leaves? Make sure you look through that, because I do want you to appreciate the different ways that they can be modified and then how we can make use of them. So for example, with stem tissue, I mean, most of us immediately, or at least I tend to immediately think about lumber for building, but there's a lot of ways that we use stem tissue. Charcoal. Is that Kingsford brand charcoal briquettes, the blue and white bag, right? I haven't looked at one of those bags in a while, but they used to have a story on the side of the bag. I was waiting in line to check out, and I was like, so I read the bag as I was waiting, and it was a story about Ford, the Ford Motor Company. And didn't they actually have a plant here in the Upper Peninsula? It's on the way to Houghton. And I'm blanking on that little area. Anyways, they used to make woody station wagons. And they were called woodies because they had wood paneling along the station wagon sides. And so I think it was Ford's son-in-law was working in it, and he saw all this waste. Once they did the paneling on the station wagons, all the rest was getting thrown out. And he's like, we should do something with this instead of just throwing it out. So they figured out how to make charcoal from it and bag it and sell it. And so, I mean, that is quite, so, he thought this was a huge waste. We should do something with it. And they came up with charcoal. So I don't know if that's still on the bag or not, but anyways, ropes, we can get ropes and fabrics out of stem tissue. If you think about linen, for example. Sugars and spices, dyes, drugs, tannins, latex, resins. So lots and lots of uses, more so than just lumber. So make sure that you do look at that idea. Oh, and cork for a lot of reasons. First, I tend to think about wine bottles, but now they do the fake cork for a lot of the wine bottles. But if you've ever wondered, they harvest it from cork oak trees. And they do it in a way that doesn't kill the tree. So the tree will produce, they can keep harvesting. And so I thought that was pretty interesting. Amber bristlecone pine, really, really old. This one's Methuselah. It's hidden. No one knows except maybe like 2 people. No one knows where it is because they don't want it destroyed by people taking samples. And then giant trees, that's not me. But you have the giant redwoods, the giant sequoias. Very, very impressive. I saw the giant, I took this picture, I think this is the giant sequoias. I went to the Sequoia National Forest in California and was amazed. Anyone gone there? Giant sequoias? Oh, you gotta go. And I can't believe this isn't me. Look at, they're on horseback. When I found out you could do that after my trip, I was just like, are you kidding me? This is the giant red. But I'm like, okay, that's next. We gotta go do that. Actually, horseback ride through the giant redwoods. So I didn't get to do that. But yeah, massive, massive, massive. Okay, so we're done. All right, so next week we'll get into that in lab. And then on Monday in lecture, I'll start with leaves. We got to get into leaves. Yeah.
Did you know that redwood wood is pretty much good for nothing but toothpicks? It's right over there.
No.